Hello friends, welcome to AKS. Today we are starting important concepts and current affairs in biotech from the past 1 year. This is part 2 of it. Let's start with gene silencing. See, gene silencing is a technique which aims to reduce or eliminate the production of a protein from the corresponding gene. That is, see, always a protein expresses itself. So you try to stop or reduce the expression of the protein. What are the repercussions of it? We will see. See, it is the regulation of a gene expression in a cell to prevent the expression of a certain gene. It is described as switching off of a gene by mechanism other than gene modification. See, in gene modification also we try to switch off the gene, but here we are trying to slow down the process, that is gene silencing. What is the purpose of gene silencing? See, the methods are used to silence genes are being increasingly used to produce therapeutics to combat cancer and diseases such as infectious diseases and neurodegenerative disorders. That is, to create medicines like therapeutics, you are trying to gene silence to cure diseases. We just have to understand what it is. See, this is normal protein production. This is the gene. In DNA, if we discuss, there is DNA replication. In that you have transcription, translation processes. See, here if you see in the normal protein production, transcription has happened, mRNA has come in, translation has happened, and the protein is expressed. But what we do in gene silencing is, after transcription, the molecules that censor out certain mRNA is removed, so they prevent translation and the protein is not expressed. There is a subtle difference between gene silencing and gene knockdown. Both are considered same, but there is some difference. In gene silencing, the expression is reduced, whereas in gene knockout, they are completely erased out from the organism genome and thus have no expression. Gene silencing is considered a gene knockdown mechanism since the methods used to silence genes such as RNAi, CRISPR and siRNA are generally reduce the expression of a gene by at least 70%, but do not completely eliminate it. See, these are some of the techniques to silence the gene. You can see CRISPR technology I have discussed in gene editing. You can watch the video for better understanding. The purpose is to reduce the expression. See, if at all 70% of expression is stopped, 30% is there. This can be used to create therapeutics. If at all you completely knock down the gene, there is no possibility to bring back it. In therapeutics, it can be used. Gene silencing can be reverted back. So, gene silencing is a very effective way to create therapeutics. Methods using gene silencing are often considered better than gene knockouts since they allow researchers to study essential genes that are required for animal models to survive and they cannot be removed. In addition to this, they provide a more complete view on the development of the disease. Since diseases are generally associated with genes that have a reduced expression, this can be useful. We have to understand that gene silencing can be used to create therapeutics. Therapeutics are vaccines, and the expression, if reduced, can have more results. Now let us discuss about gene therapy. Gene therapy is a treatment that uses patient's own immune cells called T cells along with WBC to fight against diseases. See, gene therapy is the process of using our own immune cells, that is, we have immunity in ourselves we use immunity to fight the diseases. So our immune cells are known as T cells, and this plus WBC, that is white blood cells, are used to fight against diseases. So what they do in gene therapy is the cells are removed from the patient, sent to the lab and encoded with viral vector, which are reprogrammed and returned to the patient. It is simply to say that the cell is removed and in lab they are added viral vector which are able to fight the diseases, and then again they are reprogrammed into the body. This is gene therapy. This also involves replacing mutated gene with functional gene and introduce new gene into the body to fight diseases. See, for example, a cancer cell is there which is a mutated gene. This can be removed from the body and a functional gene can be introduced into the body, and this can now fight with the diseases. There are two types of cell therapy which can be done. They are somatic cell gene therapy and germline therapy. See, this just we have to remember that there are two types of cell therapies. One is somatic cell and second is germline therapy. So now we are discussing gene therapy of cancer, that is how gene therapy can be used to treat cancer. See, this is pacified retrovirus. The next is retrovirus carrying a normal human gene. See here in the nucleus part, the cancer cell in culture infected with retrovirus. Now retrovirus attacks cancer cells. Retrovirus is fitted with a new gene. Now lymphocytes enter the cancer cells and they kill the cancer cells. So in this process we have seen what is gene therapy. That is, you use your own immune system to kill several diseases. This is gene therapy. What is CAR-T? That is, it is a personalized form of cancer treatment. If you see, the cancer is different in different stages in different bodies. So CAR-T will act as a personalized form of cancer treatment. It involves removing immune cells, modify them in lab so that they can reorganize cancer cells. You know that immunotherapy is a treatment that uses your body own immune system to fight cancer. It's called CAR-T, that is cell therapy, because it uses body own cells to fight cancer. As we have discussed, it is a personalized form of cancer treatment. It involves removing human cells and modify them in lab so they can reorganize cancer cells. This is actually the process of gene therapy, and that too CAR-T technique. See, the first patient has blood removed and WBC is separated out with the rest of the blood being returned to the patient. The harmless virus used to insert genes into T cells, that is immune cells. These engineered CAR-T cells, we have already increased immunity in these cells and we have reintroduced, and they will recognize and destroy patient cancer cells. So in this way, it is a breakthrough in treating of cancer with our own immune system. On this note, thank you very much.